Okay, good to see you again. Active transport. It's a topic we're going to deal with in this video. You can see you have a concept map that we handed out in class. The title of that map will be active transport. It will be broken down into three main concepts. The first being active transport, which we'll dive in a little bit about those foundational aspects. The second concept map will be the solute pumping, or sodium potassium pump, a topic we've talked about briefly in the past. And your third concept map will be bulk transport, and an example of that being exocytosis. If you could, on that third map, make sure you add one additional bubble. That will be E, which will continue to cover that topic as we go. So our topic of active transport, we have a couple components we need to write down. In bubble A, in our main topic being active transport, we know that active transport involves transporting substances that are unable to pass by diffusion. We've talked a little bit about diffusion and those components, but we know when we're talking about bubble A, we know this involves the transporting of substances that are unable to pass by membrane via diffusion. In relation to this, bubble B, we know when we kind of showed this in our example dealing with sugar molecules and the grading associated with those. In many cases, for bubble B, we know that these molecules or substances just may be too big to move by themselves through that membrane. In bubble C, we also know that these substances might not be soluble. So in essence, they can't be broken down to the point where they can move through that membrane. So in bubble C, they might not be able to dissolve in the fat core associated with the membrane. They might not be soluble enough to be transported through that membrane without any energy or anything else to drive it. And for bubble D, we've talked about this extensively. We know that this type of energy required to drive an active transport process occurs because that medium, that component that's being moved, more times than not is going to be moved against a concentration gradient. And we've talked about the example of a concentration gradient that helps support this component. To help support this topic of active transport, we will break it down into two main ways in which active transport can occur. The first of those being one that we're already familiar with, involves solute pumping, or based on our last video with Crash Course, we know that as the sodium potassium pump. So that's the foundational concept associated with this map. We know bubble A deals with amino acids, some sugars, and ions are transported by this sodium potassium pump. Bubble B with this topic relates to something we're already familiar with through our last video. How is that toll paid? The toll is paid via ATP. This is the process in which energy is utilized to create the relationship that moves the material across the membrane. And in this relationship, we know ATP energizes protein carriers, and in most cases moves substances across the concentration gradient. And our third bubble associated with this map deals with ATP. We know that in this process, ATP is converted to ADP, and within that combination, energy is released that drives the pump. Also, within this relationship, ATP loses a phosphate group, thus making it now ADP. We know that enzymes, which are catalysts, which help speed up this reaction, drive the reaction, allowing it to occur. To help show this relationship, we'll utilize this diagram. This should look somewhat familiar to you. We'll also make a copy of this and hand this out in class. But in our first step, here to the left, we show that sodium binds to the pump protein. That occurs here on the left. So the actual sodium it actually binds to that protein, and as we made mention of in our notes, ATP drives this process, and by driving it, the energy associated with it, and the energy associated with ATP being changed to ADP, the loss of that phosphate group via an enzyme, actually breaks the bond and creates the energy which allows this pump to change shape. And by changing shape, the toll is paid. The energy associated with that shape changing is indicated by the toll actually being paid via ATP. And as that progresses and as the shape changes, you begin to see that because of that shape changing, certain components are expelled into the extracellular fluid outside of the cell. So the shape changes and proteins expel the sodium associated with this relationship. That goes out into the extracellular environment, and on the outside you have potassium components that actually once again bind with the protein. They bind with that pump-based protein, and those potassium components, and as we said, once potassium binds to these proteins, that causes the release of the phosphate group and that energy exchange to be complete. We know the next component here to the right, you can see the relationships that are now occurring. We have potassium that still is involved with that protein, and the loss of that phosphate restores that protein in the pump to the original shape. And once that occurs, the bottom portion, as you can see here, opens up, and that potassium component is released back into the cell, into the cytoplasm. And from this point on, sodium once again is ready to bind to the protein again and start the process all over again. The second type of active transport process we're going to talk about involves bulk transport, and we'll hit this again in the next day or so, but an example of bulk transport is exocytosis, and we'll support this with a diagram as well. Just basic components. We know this process moves material out of the cell. It's a process in which material is carried in a membranous vesicle, and if you remember from Golgi apparatus and rough ER, we know vesicles move components within a cell. We know that these vesicles migrate to the plasma membrane, the outside region of the cell. And this process involves the vesicle actually combining with the plasma membrane. And once that's done and that combining is complete, material is emptied on the outside portion of the cellular membrane. And this diagram will help explain that. Once again, this will be something we pass out in class tomorrow, but you can see the relationships that take place here. You have material being carried through a membranous vesicle, and remember, vesicles are those types of things that transport material within a cell. We know that those vesicles move to the plasma membrane or cellular membrane, and in doing that, eventually they come into contact with that membrane and actually combined with it. And doing so, after that relationship takes place, the material is eventually emptied into the extracellular environment. And if you remember from the Golgi apparatus, this is very similar to one of the pathways we discussed in those diagrams, where proteins were actually moved from the Golgi to the cellular membrane and outside of the cell. Okay, so to finalize, we've talked a little bit about active transport. We talked about the sodium-potassium pump. We've talked about bulk transport via exocytosis. We'll have a couple diagrams to work with in class tomorrow. If you have any questions, let me know. Thanks.